So overall, the general pathway for this is that you have your ears or your spiral ganglion cells in your ears that go onto your brainstem where there are neurons, and that goes onto the thalamus and then the auditory cortex. She stresses this a lot in the next few lectures, but this is generally what happens. You have the sensory organ and then the brainstem and then the thalamus, and it goes to the respective area in the cortex. So tonotopy, the only thing you really need to know about tonotopy is pitch and intensity. It's like this different way of encoding or organization in the cochlea that helps us determine sounds or understand perceived sounds. So with pitch, what it does is that it activates different parts of the cochlea. And the two parts or the gradient that you see in that is that you have an apex and a base. And then it's the same in both cases where the apex things start low, base things are higher. So with intensity, it's just the whole thing is activated, but it's activated less or more depending on the amount of how like strong the intensity is. And so if it's more strongly, there's more action potentials going on. So pitch is specific, intensity is like how intense it seems. And then based on what we learned from tonotopy, this is how they developed the idea of cochlear implants, that you can manipulate what you hear if you can somehow have a device that really helps you. It uses like electrical signals to affect or to like fake the pitch and intensity for people who can't hear, so that it would still send the neural processings that you would hear then. So that's it for tonotopy. Moving on to plasticity. So there are 3 steps to this. So with plasticity, you can hear something as a kid, and that's like pretty important. It's not completely hardwired, but it's your tonotopic map in your brain is rather experience dependent. Meaning, if you hear a second language as a kid, it's more ingrained into your mind or into your brain versus if you heard it in adulthood, it would not have the same effect on your tonotopic map. Instead, during adulthood, there is some change to your tonotopic map, just not as much. The main point for adulthood is that if you hear the same sound again and again, there's less activation in your brain then. But if you associate that sound with a reward, then there's not so much habituation going on. You'll still have a lot of neurons be turned on by that sound or by that reward, or well, by the sound associated with the reward. And then lastly, you have a moment by moment basis, which is just talking about alertness. The thing about alertness is just that you'll learn about this again in attention, but as alertness increases, your responses increase. Okay, so somatosensory system organization. The receptors are called mechanoreceptors. They're basically like everywhere on your skin. And so they have these ion channels that are dependent on the stretching and tension of how you're moving yourself, really. So the process behind that is that you have sensory information that goes to the dorsal root in the spinal cord, and then that projects onto the CNS. And then that's basically just saying, like, this is regurgitating it and saying that it's the information from skin is relayed onto the brainstem, which then again goes to the thalamus, which then goes to the cortex. There's the point of 2-point discrimination, which is the activity you did looking at your like tactile acuity. And the reason why for some people or for some areas of your body, if you have 2 fingers touching it, you can either tell it's 1 or 2. That distance depends on the convergence of your primary sensory neurons and into a secondary neuron. So if you have a lot of primary sensory neurons that converge into a single secondary neuron, if you have a lot of those, then you have a wider receptor field. But if you have like only like 1 or 2 converging, there's less convergence, so there's going to be a smaller receptor field. So you have to have 2 fingers be closer in order to detect it as 1. This is the homunculus. This comes up again with motor systems, but the point of this is that some areas are more sensitive than others because there are more nerve endings there, and it's better represented in the brain. It's just like how much of your brain is dedicated to certain parts of your body. And then there's the somatosensory one, there's the motor one. So somatotopic map, which is essentially what we see here, this map that you see in your brain. What happens in the case of amputations is that let's say you lost like a finger here. Let's say you lost your middle finger. So this middle finger area would be decreased. It would be a lot smaller in your brain. And then the areas on your brain for the rest of your fingers would kind of slowly take over that area. So the last four fingers will become more sensitive in place of the missing finger. And then that's what causes you to have the sensation called the phantom limb sensation. So then, like, you're missing that middle finger, right? But if you touch like your ring finger, it's so close and it's like moved so much, it's grown so much into the area of your middle finger that it'll feel like your middle finger is still there, or that you're touching your middle finger despite it not being there. So that's phantom limb. And then the last part of this, oh, I don't know, I have an extra slide, but that's the last part of it is pain. So in the case of pain, the receptor for pain is called the nociceptor. So nociceptors they're basically you just need to know that it's mechanically, thermally, and chemically stimulated. That's all I think you really need to know about that. Moving on to motor systems. So your body movement, she asked on the lecture or on the study guide that you know what Lockton syndrome is. And with Lockton syndrome, that's like you being fully awake and fully conscious, but you also can't move. Like you can't voluntarily move. I don't think she really expects you to know to understand what's the reasoning behind that. Just know what it is. And then with skeletal muscles, you use your skeletal muscles to voluntarily move. And then that has to do with motor neuron axons in the spinal cord. This goes back to the idea of topographic mapping in your brain, which is like the homunculus I just showed you. And more so what I mean by topographic mapping too is that there's just basically different areas in your spinal cord that respond to your feet. Like not if you move your feet or anything, you're not going to feel it at the very top of your spinal cord. You're going to feel it closer to like the bottom of your spinal cord. That's the topographic mapping, which also maps again in the brain the same way. The neurotransmitter you need to know for this, you don't need to know anything other than just what it is acetylcholine. So if there's anything related to motor function, the neurotransmitter you're looking for is acetylcholine. That's it. There's also graded contractions. Again, it's like the concept of not a full action potential where it's just, does it happen, yes or no? It's to what extent does it happen? To what extent do you feel it? And then there are three types of inputs with body movements, and we are going to go into those three. So the first one is sensory feedback from muscle, and then the receptor for that is proprioceptors. And what proprioceptors do is that they detect the stretching of your muscle. And then based on that, they directly synapse onto motor neurons. So that's the idea of a stretch reflex. So the example for that is the knee-jerk reflex. If you take something and hit your knee, it projects like that stretching. It projects it such that like, if you can imagine your knee getting tapped right now, there's both the like part of your leg stretches and part of it doesn't stretch. It's like this cycle that projects from your motor, like your actual body, through your brain back down to your body. I feel like there's just like you should just know like this definition and you should be solid. There are quite a few reflexes here, which is why I don't want to confuse you too much and just keep it straightforward. So the next thing is local interneurons in the spinal cord. And then you have something called antagonistic muscles. And this is the idea of flexors versus extenders. So you have something called reciprocal inhibition. So with reciprocal inhibition, what's going on is you have, I'm so sorry. So you have like your proprioceptors activating this innervating neuron, and then what that does is suppresses the antagonistic muscle. So it's just like this idea of a plus and a negative. It's always coupled such that one thing flexes, the other extends. And that brings us to flexor reflex.